Our first speaker is David Colameo from University of Zurich. David is senior scientist at the electrophysiology core facility in Zurich, and he's developing high throughput and high content techniques that combine automated patch clamp and advanced imaging.
Yeah, good morning, everybody. Thank you very much for the invitation to talk here, and I'm excited to present some of our advances using this technology, great technology that we have in our lab. So before I start, I would like quickly to introduce also our facility, because it might seem a little bit strange that there is the need for an electrophysiology facility at the university. So we are part of a bigger consortium of core facilities at the university, and we provide full support from assistance from the beginning, experimental design up to also data analysis and data acquisitions. We can also train you to use our instruments. So you can book it individually and people can become really independent. And we can also talk about collaborations. So from the method side, we have like 3 pillars. We provide traditional patch clamps. So my PI, David Benton, who couldn't be here, unfortunately, he's a really talented patch clamper. We also provide network activity assessment in neuronal populations or cardiomyocytes using HT-MIA and calcium imaging. And of course, we're also proud owners of SynchroPatch 384 in our lab. And I think we are one of the first institutions, at least in Switzerland, to have this machine. And yes, so apart from that, we also try to really push kind of the boundaries of what we can do with these machineries and see what do our customers actually want. Now, I don't need to kind of convince you of the advantages of having high throughput electrophysiology here, but just very quickly how this machine works is that you have cells in suspension, and then you can, in a high throughput manner, patch them in a planar kind of way. And we were also quite successful in trying also different cell populations. So here, for example, you can see action potentials from neurons using this machine. And this GIF was actually made by Tom in the first training session that we ever did here at Nanyang. Still great to use it for presentations. Yeah, but as you may maybe know, like in the academic field, sometimes it's difficult to convince people for new technologies. And one question that always comes up is like, how about heterogeneous populations? People are working with not always HEK cells or very homogeneous cells. And so one question that we always had is like, how can we identify cells in using automated patch clamp? Now, in order to tackle this question, we wanted to use like a system that we knew very well. And this is the activity of this specific potassium channel that is very important in kidney physiology, where it's used for potassium homeostasis. And what's really neat about it is that we had already cell lines in our lab where you can have an inducible cell line, which usually doesn't express ROMK. And then upon doxycycline, we have then expression of ROMK. So what we did is to generate artificially 2 populations, having one expressing stably M scarlet or being red, and the others being green, and just induce one group. And then how we designed this experiment is to have on our well plate setup distinct groups where we can look and analyze first uninduced cells, induced cells, and then have like a mixture of the 2 populations, and then try to see if we can separate those 2 populations later on. What we also did is to stain directly for ROMK to control for the expression and induction of this protein. Now, electrophysiologically, we can use these measurements as a ground truth because we exactly know how they behave. So we can measure reversal potential, which is like an approximation for the resting membrane potential using voltage clamp. And so basically for the non-electrophysiologists, we can measure at which point the current is 0, which then corresponds to the reversal potential. Now we know from literature and also from our experiments that cells that express ROMK, they are hyperpolarized, they have an increased sensitivity to potassium, of course, and these currents can be blocked by barium. So before I go on, I would also quickly to introduce some of our other innovations, which deals a little bit with the management of these big data sets. So we are grateful about data control, but we soon realized a little bit the limitations of this. So we created like a unified R package, and this was inspired kind of by multi-omics data sets that we also have regularly in our lab, where we create one unified object, like one kind of table that is multidimensional. This just allows us to organize our experiments in different tables. Basically, we have, for example, one table explaining the sweep conditions like voltages and liquid periods and so on. Then we have the column table where we can describe kind of like the experimental conditions of the cells that we patched. And then we have, of course, all the numerical values, actually online analyses and QCs that we can measure. And so we have like this multi-dimensional object that makes it really easy also to filter out and combine data. And yeah, so of course, not everyone in the lab is really adept to programming and scripting, such as my PI here. So we also developed a web application to kind of like manipulate these objects online and then visualize it and perform different calculations also without using a scripting language. And of course, we are always interested in having beta testers. If you're interested to maybe try it out, you can contact us and we can see what we can do together. So yeah, coming back to the experiment, what happens when cells are induced and have ROMK expression? So the biggest change that we see between the groups is, of course, this change in reversal potential. So we have here, I'm sorry about the colors, they are like automatically chosen, but we have here the group of M scarlet, which is not as hyperpolarized as, for example, induced ones here, the GFP expressing ones. And then we have the mixed populations that is kind of like in between the two. Something else that we see is, of course, in different liquid periods, if we add like a high potassium solution extracellularly, we have this influx of current. And maybe a little bit anecdotal, as an anecdote, when I first did this experiment, I thought like this experiment was failing because my catch was super bad after the whole cell configuration. But it turned out that this is kind of a feature of cells expressing this channel because it's basically an open pore selected for potassium. So once you actually add here barium, it plugs up all the ion channels and the seal reaches kind of like the normal levels. Yeah, so if we quantify kind of like the induction efficiency, we see that, of course, the GFP groups that were induced with doxycycline are the most depolarized together with the mixed one compared to the M scarlet. But there is also some kind of also biological variability to the system. And this can have like different reasons, which is inherent to biological systems. So we can quantify kind of like the number of cells that are hyperpolarized and have like an idea of our population level induction efficiency in our groups. Like, we also wanted to explore ways to how we can kind of make use of this heterogeneity in our cellular systems. And we thought of using dimensionality reduction to kind of like try to cluster cells that have a similar response. So what we did here is kind of feed all these three matrices in an unbiased way, and we did principal component analysis and then some clustering to see if groups could be grouped by functionally similar ways. And indeed, we see this like two big clusters, so you can choose as many clusters as you want, but here in this case, like two clusters are quite nice to have. And so we can then group kind of like cells by these two clusters. And we see that this method identified hyperpolarized or non-induced cells versus the depolarized cells that are not induced and the depolarized ones. And here in the MS Scarlet one, of course, there were just a few ones that, yeah, exactly. And then if you visualize the reversal potential in this group in the clusters, we see that it's kind of like a gradient of this of the EREF that we see. And they match quite well also the induction states that we just say, okay, we just put a cutoff of -50 and they separate quite well between the two. Now, after that, after the experiments, we of course don't throw away the plate. This is like one of our biggest assets at the moment, so we keep them. And we also leveraged the liquid handling properties of the biomech, yes, to perform fixations and immunofluorescence like in the synchropatch itself. So we fix the plates, then we go to the spinning disk at our microscopy facility and perform high content imaging. So we scan well by well. This is also all semi-automated, so it goes well by well, and we can image the whole. So we take one image in the bright field. What we then did as a routine, we segment this bright field image, and then we overlay it to the fluorescence. And we can have then a multivariate quantification of each cell. So we can have information about the pixel intensity, the size, and the shape. And you can appreciate kind of like what the cells look like after patching. So we see these two groups of cells in green and red. But on top of that, we also did this immunofluorescence against ROMK itself. And we see maybe, I hope you can appreciate it, that only the green cells have like this ring of gray ROMK expression. So we wanted to quantify that. So we can, here it's a little bit bigger. We wanted to quantify the number of cells that actually contained, that expressed ROMK, like in the general population, the image. And we found that it's nicely correlating with the electrophysiology. Like if you look at the proportion of hyperpolarized cells that we measured in the different groups. And the big question is, can we identify the patched cell? So here you see the results of the segmentation. So this is most probably the cell that was patched. And we can then segment and measure different values that are inside this hole, such as pixel intensity, area, percentage, and so on. And here, as you can see from the cumulative distributions of the three channels that we have, like in the green, red, and far red channel, we see that the separation is quite nicely between the two groups, here in this case with GFP and M-scarlets only. Some of the challenges that we are kind of facing is that, of course, these are patched cells, so they went through a lot of troubles. Sometimes we lose the fluorescence just because like fluorescent proteins are quite small and can leak out. But still, we wanted to see if we can kind of identify the cells and then classify it and correlate it with the electrophysiology. So there are like different classification strategies that we can take. For one, we can just be threshold based and say like, okay, 0 up, or if it has this value and so on. Or we can also use just a multinomial regression based system where we just use the pure population to learn how like a patch cell should look like, and then try to classify it in the mixed population. Now, again, like here to reiterate, we just use the pure conditions to learn about the patch cell. And then when we use these two values, the pixel intensity and the area that is covering the whole. And these are the results that it comes up with. So first, like the ones that were predicted as GFP positive, see that most of them are lying either here, and some outliers in the M-scarlet group. And the ones that were predicted as M-scarlet, we have a few that were wrongly predicted to be GFP, and but most of the M-scarlet positive ones are really predicted as M-scarlet. We have, of course, like also two ground truths. For one, we know that these two are mislabeled. And secondly, we can use also the electrophysiology to kind of have an idea if it's true or not. And these are these these groups here. So basically, hyperpolarized cells cannot be M-scarlet positive. So to kind of benchmark like the efficiency of our model, we can use these two ground truths. For one, the experimental condition and the hyperpolarization state of the group to measure the sensitivity and the specificity of our assay. And using that, we come up with a pretty good number for now. So we coming up with about 90% for depolarized cells and 80% hyperpolarized ones, showing that the sensitivity and specificity is quite nice. Of course, we're trying to push always for more, but for now, this is what we came up with this system. Now, after having established this pipeline, I wanted to also give an outlook on what we can do with this system. So, if time allows, I really quickly want to go through one of the projects that we have. So we heard a lot on lysosomal storage diseases, and we are collaborating with this group that generates IPS lines, patient-derived IPS lines, from patients that are suffering from Sanfilippo syndrome, which is a rare disease that results in accumulation in heparan sulfate in lysosomes and a lot of different problems, including childhood dementia. So what they can do is have like the IPS lines be differentiated in different cell types, including excitatory neurons, inhibitory neurons, and astrocytes. So what we did here in collaboration is to mix them together in a triculture system and then try to look at the development of these neurons in terms of electrophysiology and in general development. So first off, we wanted to look at the time course of maturation using our HT-APC system. So here you can see population level activity using this chip arrays. So basically this is the spatial distribution of the activity of neurons. And here you can quantify also the activity over time. And what's really interesting about neural cultures in general is that they tend to form this burst activity, so this synchronized burst activity, which is dependent on synaptic maturation and connections between the neurons. So we can also visualize it over time. So this is like repetitive measurements from the same cultures over time and compare it then to our Sanfilippo lines. And we see that this fail to kind of have this synchronized state. And we can quantify it over time, and we see that compared to wild type cell lines, these Sanfilippo ones have great impaired network activity. But doesn't mean that they are not active, since at the very beginning, especially, we see this individual activity that is very high compared to the wild types. Like in terms of electrophysiology, neurons are a little bit more interesting to patch than ROMK cells. So we can look at inward and outward currents and quantify it in IV curves. And very early on, this is like an early measurement that we did when we were setting up the system. We found that we had like an intrinsic heterogeneity in the responses of our cells. So there were some cells that had like this clear sodium inward rectifying sodium currents, whereas like the probably astrocytic fingerprint where you just see this outward currents. So in order to kind of tackle the question whether which cell types we actually were measuring, we used this AAV approach. So we used a combination of three AAVs that were expressing three different fluorescent proteins under specific promoters, like DLX for inhibitory neurons, CAMK2A for excitatory ones, and then GFAP for astrocytic. This is how it looks like after the patch. Again, we can identify the whole and then see and try to see what was the identity of the patched cell. This is very preliminary for now. We are still repeating these experiments, but we see that there are some groups of cells that express only one or the other, so there's like a clear segregation. We don't know yet what we should do with this ambiguous cell population. So for now, we just group them as separate. But if you lay on top like the electrophysiology, apart from the ambiguous one, like the astrocytic, they actually are quite different from the ones that we predicted to be neuronal with this clear inward rectifying currents. So just to summarize what I presented to you today, I hope I could convince you that looking at population level matrices might be also a good option to use this technology. I showed you like the correlative imaging pipeline where electrophysiology and imaging can complement each other. And we are also getting more and more to complex cellular systems where we can look at iPS-derived models as well as primary neuronal and tumor samples. So we have also projects on adenomas in the adrenal gland. So we are quite interested to push these technologies. And yeah, so I'm coming to the end of my presentation. I would like to thank, of course, collaborators, lab members, and everyone that contributed to this project.
Thanks so much, David. Super interesting talk and new application combination of APC and imaging. Any questions from the audience?
Thank you, David. Fantastic talk. I'm really amazed by what you do. With respect to the fluorescence, would you also be like a technical question? What is the resolution? Can you also detect like intracellular fluorophores or something like that? That you have the calcium fluorophores. Let's say you have release of calcium in the cell. Can you detect that? Would be able to detect it from intracellular signals?
So you mean you have like a GCAMP expressing cell and then look electrophysiology? That would be super cool to have, but unfortunately it's a different machine that we use for the imaging. So we fix the plate and then we go to the microscopy, right? So we don't have like capabilities to but yeah, I don't think that's technically would be quite challenging to do. Maybe you could try it with unfixed cells and hope that the cells are still living, but unlikely that they will survive the whole process after washing out the internal solution and everything. Yeah.
That was a lovely talk, David. So something that's bothered, I think, a lot of automated patch clampers because they're doing it in the blind is how many single cells you get and how many of those wells you might have little doublets or clusters of cells. So what did your imaging show in your plates?
That's a good point. And this was actually also the reason why most of the mixed populations were wrongly annotated, because in the very first experiment, we just wanted to be sure we just threw in all the cells that we had. So we had like a high concentration of cells. And of course, we had a little bit more overlap of other cells that were not patched in the vicinity of the hole, which then probably were classified wrongly. And so what we did for the subsequent experiments is to really titrate down the cell number to have like still a good catch rate, but not to overwhelm kind of the vicinity of the hole. What we also found is that sometimes having like a ligand puff cleans up the area quite nicely. So with neurons, we sometimes also do ligand puffs. So you have like the patched cell there that still stays, but all around there's nothing. And yeah, it also depends a lot on the type of chip that you use. So we were using a lot of expired old thin plates at the beginning because we couldn't afford more. So the hole was much bigger than the newer ones. And of course, then if the area of the hole is bigger, then you might have also like these confounding factors of having other cells identified. So really try to troubleshoot all different scenarios here. Yeah.
Thanks, David. Great presentation. Let's thank David again and move on.